Let's just study about number, size, and concentration. If you have a really big number like this, then at once it's difficult to recognize how big it is. We need to count the number of zeros, right? So to make it easy, we wanted to make some package like 10 to the 3,000. And this can be continued, and another 10 to the 3 becomes million, and then another 1,000 is a billion, and then trillion. So in Western culture, and they use this kind of a 1,000 unit, 10 to the 3 as a package to make the bigger number easy to recognize. Well, in comparison, in Asia, we have a 1, 10, 100, 1,000, and then 10,000. Interestingly, this cycle is in the 10 to the 4, man are the unit. And in English and in Western culture, it's 1,000 unit, but in Asian culture, that is the 10,000 are the unit. So after man, it becomes ok and jo, which is commonly we use. It can go further, maybe gyong, he, si, but after this, maybe usually we don't use. And the last one, guk, is extremely big. The Chinese character means that's a 10 to the 48th. So this system is kind of East Asian, the culture, Chinese-based the numbering system. Then what can be the biggest number? Maybe in Indian culture, the send particle number in the river named Hanga, that is assumed that's a 10 to the 52. And Bulgasai, that means it's difficult to imagine, right? That is 10 to the 64. And maybe the biggest number in Indian culture or Buddhism culture is uncountable big number, that's a 10 to the 68. Well, how this number is calculated, that's not very clear, but this is for fun. So this is kind of representing the big number. Now, coming back to the Western culture, if we continue, so after billion, trillion, quadrillion, 10 to the 15, so now we see some rule here. So after million, billion, we can feel like a number 2, trillion, number 3, quadra, number 4, then it keep going, like a quintillion, and sextillion, and septillion. Octillion is easily we can catch, that's some number 8 in there, and then nonillion and decillion kind of things. Now it can keep going with this the numbering system. Now the biggest number in the website you can find is maybe Google. Google is a 10 to hundreds. Of course, you can make it even bigger with this. 10 to the Google is maybe even much bigger numbers. So anyway, this is the system that we wanted to make the numbering system is easy to capture. If we learn a new language, one of those basic things is the counting of number. In English, it's a 1, 2, and it goes on 3, 4, 5, 6, 7, 8, 9, 10, and we just have learned that how we can deal with very big numbers, right? 1,000, million, billion, or trillion. Inside that unit, we use this numbering 1 to 10, and then we can count the numbers. In science field, we have a different way of the counting. So here is the one system. 1 is mono, 2 is di, and 3 is tri, and 4 is tetra. If you have one substituent, we call mono, or sometimes we don't use it. It's obvious. If just a simple single one, then count or no count. But if it's a two substituent, now we need to count it. So for example, this is adenosine diphosphate. There are two phosphates here, so you cannot just simply say adenosine phosphate. You need to give the numbering of the number of phosphate here. If it becomes 3, then this is ATP, adenosine triphosphate. So this is quite unique and also useful way how to designate number of functional groups. Now in this case, the phosphate is connected to each other and we call mono, di, tri. And if it goes further, maybe 4 is a tetra. How about this case? This is the inositol with 3 phosphates. Now, do you see the difference between adenosine triphosphate? In adenosine triphosphate, the phosphate is connected to each other. But on the new other example, inositol phosphate, now the phosphate, there are 3 phosphates, but they are not connected to each other. They are attached to different positions. Then, if you call this as inositol triphosphate, now it's a little confusing whether the phosphate is connected to one position together or it's separated, scattered in different positions, 3 of them. To avoid the confusion, now we have a little modified version of counting. So 1 is still mono, and 2, instead of a di, we call bis, and 3 is a tris, 4 is tetrakis, 5 is pentakis, and here's the example. Now here, inositol with 5 phosphate, but those phosphate is not connected to each other. They are scattered in different positions. So this is called as inositol pentakis phosphate. Now how about this? It has 6 phosphate, right? Then this one we call inositol hexakis phosphate. Now, in organic chemistry, the basic molecules are organic molecules, which means the molecule has the connected carbon scaffold. So we give the number of carbons in the molecule, and then to designate that number, we use still different system. So single is metha. So methanol's metha, right? And then two carbon connected together, it's an ether. So this is ethane, ethanol kind of cases we can see. 3, propa, and propane, propanol kind of thing. And 4 is buta, 5 is penta, 6, hexa, 7, hepta, 8 is octa. Octa we have seen, octopus. The octopus has 8 legs, right? So that's meaning of 8. 9, can you guess? Is it nano? No, it's nona. 10 is deca. 11, undeca. 12, dodeca. So we can continue this way. And 13, trideca. Easy, right? In spectroscopy, if there are the many peaks there, then we have a still different system to count how many peaks are there. So single peak we call singlet. And then if there are 2 peaks, then we call doublet. And 3 peaks, triplet. 4 peaks, quartet. 5 peaks, quintet. 6 peaks, sextet, not hexatet. 7 peaks, septet. 8 pigs, octet. 9 pigs, nonet. 10 pigs, dectet. How about more than 10 and there are many pigs? It's difficult to count and overlapped to each other. It's difficult to assign the exact number. How will you handle? In the case, we simply say there are many pigs, multiplet. In computer system, we are commonly hear this term of binary. So binary means it's the meaning 2, and this means the single unit of information. One bit is composed of 2 information, 0 or 1. If that has just a single single information, maybe you can call it unary. If it is a 3 information, 0, 1, and 2, this can go ternary. If it's a 4, it's a quaternary. This is important because our biological information system, in DNA, we have A, T, C, G, 4 informations in 1 bit. So, computer system, if it is binary system, our biological, the information system is a quaternary system. Okay. Now, if we keep going, then 5 is quinary, 6 centenary, 7 is a septenary, 8 octenary, 9 nonary, 10 denary. So denary is in our term, that's a 십진법. So that is our basic numbering system in most of those the modern other cultures use as kind of a numbering system. So this is another way of counting the numbers. Let's just think about the scaling in science field. Our standard, the measurement unit, is a length-wise it's a meter, and weight-wise it's a gram, and concentration-wise that's a molar concentration, is standard the measuring system in science. If the number becomes very big, there are too many zeros, it's difficult to count. So to make the package in Western culture, we have seen thousand, million, and billion and trillion kind of the scaling. Now that scaling in science field, we have a little more systematic way. If it becomes 1,000 times bigger, then we put large K, so that is a kilo. 1,000 times bigger, that's a million. Now we put M, and that's a mega. Next one is a billion, right? In billion case, we give the name as a giga with a symbol of a large G. And trillion, that's a tera, so which is we commonly use in computer memory scaling. And keep going, we get peta. Now we are in the middle of peta and the tera range. Our current the hard disk size is several terabytes right now. In several years, it may become peta. If it goes higher, then after peta we have exa. And then next one is zeta. So we will expand our scale bigger and bigger in the future. Now this bigger number, everything is written in capital word. Capital are the alphabet, the big alphabets. Now if you go down, smaller size, now 10 to the minus 1, 1/10 is deci. So small d we use here, which is not very popularly used in the science or chemistry for now. But 10 to the minus 2 we use quite often, centi, that's a centimeter. And then 10 to the minus 3 is a small m, milli. So this is very common we use. So millimeter, milligram, millimolar kind of units we use. The next one, now packaged as 1,000 times smaller, 10 to the minus 6, that is million times smaller, that unit, micron. So here, for convenience, I just used the English alphabet as a small u, but actually this should be Greek word, Greek alphabet of micron, mu. And 10 to the minus 9th. Okay. Do you know what that is? 10 to the minus 9 is nano, not nona. Okay, nano is a smaller side, nona is a bigger side, 10 to the 9th. The next one, 10 to the minus 12, that's pico. 10 to the minus 15th, that's a femto. 10 to the minus 18th, that's atto. Currently, we commonly use from micro to femto range in chemistry field. Atto, rarely we use. The even smaller one, maybe it may need some more time. We expand our scope into even smaller at the scale and word. Now, utilizing these units, instead of saying big numbers or very small numbers, for example, 0.0000 something, it's difficult to catch exactly what's the number. So then by using this kind of a prefix, we make those numbers easy to recognize. Here is the useful summary of size of objects. Human is, let's say, about 1 meter range. And then eggs is about 1/10 of that. Chicken egg is even smaller. Ostrich eggs is a little bigger. That's several centimeter to 20, 30 centimeter size. And then going down, frog egg, fish egg, that's about 1 millimeter size. Human egg is much smaller. It's about 1/10, about 100 to 150 micrometer size. And here, importantly, the animal cells, plant cells. The common cell size is approximately 10 micrometer size. Of course, a little bigger cells maybe 20, smaller cells they are maybe 5 micrometer, but that's good to catch the cell size approximately 10 micrometer range. Bacteria is 1 micrometer at the size. Mitochondria is also originated from ancient bacteria, we believe. Mitochondria or the chloroplast, they are also in the range of a bacteria size of 1 micrometer. Now, virus. The average or the common size of a virus is, again, 1/10 of a bacteria. That's about 100 nanometer size. That's easy to capture, right? And then molecules is now, they are in the nanometer size. And one atom is approximately 1/10 of it. That's 0.1 nanometer size. So atom size is a 0.1 nanometer. That's why we have another unit mimicking atoms A with a small circle on top. That's angstrom. So 1 angstrom is 0.1 nanometer. If you look at the size one by one again, human egg cells. It's the biggest cell in our body. It's a 150 micrometer size. Sperm cell is much smaller than that, and maybe the total length, including the tail, flagella, that's approximately half size of an egg, about 60 micrometer, but head size is even smaller. Macrophages, or the all other cells, the animal cells, usually they are 10 micrometer. And lymphocyte, a little smaller, is about 5 to 8 micrometer. But anyway, those average or the approximate range of the cells, we can say about 10 micrometer. RBC is even smaller. The red blood cell, that's approximately 5 micrometer. Bacteria, I said, that's about 1 micrometer. That's good to memorize. And mitochondria is similar to bacteria, so that's also in the range of 1 micrometer. Virus, 1/10, 100 nanometer. Microtube is one of the cytoskeleton in the body. The diameter is about 25 nanometer. And protein. One example is the immunoglobulin. This is a basic structure of antibody. That's kind of this dimension. The length, the Y shape of this molecule, length is approximately 10 nanometer. And width is about 4 nanometer. This is very good to know. Now, cell membrane thickness is about 4 nanometer. And one molecule, C60, buckable or fluorine, this is about 1 nanometer size. Now we are getting into atomic size. That's why we use this angstrom unit. 10 angstrom is 1 nanometer. And the molecule, porphyrin, is a relatively bigger molecule. That's near to 1 nanometer size. Small molecules is now it's in angstrom range. So 2-3 angstrom is the small molecule size. Atom is also maybe 1 angstrom, a little smaller or a little bigger range. How about the nucleus size? Nucleus is much smaller, 10 to the minus 15. What is the unit of this? That's femto, right? Nucleus is femtometer size. And surprisingly, comparing to the atom size, nucleus, the radius is 100,000 times smaller than atom. So that means in atom, the heaviest part is the nucleus, but that occupies very small other space. All other space is filled with electrons. Now, let's practice a little of this scaling system. Let's say this is computer memory, 1.5 by 10 to the 10th byte. Then, how would you convert this into more convenient the numbering system? Okay, this is a 15 gigabyte. Giga is a 10 to the 9th, right? So that's why if you convert this, 1.5 becomes 15 and gigabyte. So if you compare these two numbering systems, it's obviously the 15 gigabyte is much easier to recognize. So for human being, maybe between 1 to 10, or at best maybe 1 to 100 range, this may be easier unit to capture. That's why instead of saying 1.5 by 10 to the 10th, so we wanted to use this simplified version, 15 gigabyte kind of system. How about this? 6.2, 10 to the 12 byte. This one, 6.2 terabyte. Easy, right? Okay, so we are studying science, so let's think about concentration and length. How about this? 0.1 meter. That's a 10 centimeter. As commonly we use in daily life. How about this? 0.001 meter. 1,000 times smaller than meter. We have seen that's millimeter. So this is a standard scientific unit. How about this? 0.00012 meter. Difficult to say, difficult to catch the exact number. This one, 120 micrometer or 0.12 meter, 12 millimeter. So I would prefer to say 120 micrometer for this. How about 0.00012 centimeter? That's 1.2 micrometer. Meter, centimeter, that's 100 times. So 120 becomes 1.2. How about this? 0.005 micrometer, 5 nanometer. It's much easier to capture, right? This one, 160, 100 nanometer. This is 160 micrometer. Now, let's think about the concentration. This one, 10 to the minus 6 means this is micro, million times smaller, right? Then this becomes 1.5 micrometer. How about this? 10 to the minus 8, you can convert this into 10 to the minus 9, then that becomes nanomolar, and this becomes 37 nanomolar. How about this? 10 to the minus 13, maybe 10 to the minus 12 is a pico. You can adjust this, then you can figure out this one is 5.6 picometer. As you see here, in chemistry, we deal with molecules. One atomic size is a 0.1 nanometer, and the molecules easily becomes around the nanometer size, and maybe proteins and other bigger the machineries in biological system, they becomes maybe tens of hundreds of the nanometer size. So that's why converting this very small number into very the familiar number in terms of a nanometer size or micrometer size, this is quite important and make you easy to capture the size of the sense of size from your object of study. In the same way, the concentration of the molecules in the body, especially drug molecules, biological molecules, is time to time super high, millimolar concentration, the range, but the drug molecule going into our body, usually they should not be that high because too high exogenous molecules, potentially they can be dangerous. So that's why those molecules in the body, usually in micromolar concentration or nanomolar concentration, and ideally the smaller the better, maybe picomolar concentration is kind of a target we want to achieve. That's why converting all the units in micrometer, nanometer size in chemistry and micromolar, nanomolar, picomolar kind of concentration range is quite the useful unit you need to learn.